system physiology. Today we are gonna start the first part of the GI physiology, and let me tell you something. Um, in my experience, since I've been teaching in PA programs, we always receive a report from the NCCPA telling us which are the weak points that the students, for example, when they go to the PANS, they send us the report so we know which topics are the weakest. And digestive system disorders, okay, is the one that always has the big problems. And I think that the reason is easy to understand. The digestive system has many different organs. Okay, if you start with the mouth, the esophagus, the stomach, then the small intestine, large intestine, and then you have to consider the autonomic nervous system part, also the humoral part with all of the hormones and peptides that regulate these, and the many types of diseases that affect different areas. So it's a vast problem. And then you have to add the nutrition part to this. So it's very complicated. And the cancer part, many different things. So it's something that has to be, I always tell this to the students so they know where they have to make emphasis. I know that many students, when prepare for the PANS, they emphasize cardiology, pulmonology, and the pharmacology, of course, but this is something that also should be considered. So this is the first part, and we are going to be talking today about the big picture of the GI tract. This is an overview of the GI function and regulation. Okay, in the next encounters, we are going to be talking more specifically about motility and then about the functions of the liver, which is a vast topic. Okay, so we are going to be trying to see how the structure and the function of the different organs relate to each other. Okay, and what is the relationship between these functions and the absorption of nutrients, assimilation, then immunity, excretion of different materials. Remember, the GI tract is one of the most important portals of entry of different pathogens, so it has to be very well protected. We are going to be talking about the secretory part of different areas of the GI tract. And also, we are going to be talking about different regulators, neurotransmitters, peptides, etc., and how they work. And of course, the regulation by the enteric nervous system and some local responses. Okay, in the second part, we are going to see mechanisms that allow the nutrient delivery, how we break them down for absorption. And we are going to be talking about nutrients, carbohydrates, for example, fats, proteins, and the role of different molecules, different substances, for example, bile, pancreatic secretions. We're going to be talking about vitamins, minerals briefly. This is not a lecture that is specifically nutrition, but simply knowing the most important basic things that we need to know about this. And also, we are going to be talking about the regulation of food intake. It's a complex regulation that may lead to different nutritional disorders like obesity or sometimes anorexia, etc. So this is a brief overview of the different functions of the GI tract. Notice that I have highlighted some because are the ones that we are gonna discussing. Okay, more importantly, digestion, of course, the most important one, breaking down the food, then all of the secretions that are necessary for this digestion, motility, propulsion of the foodstuff from the mouth to the anus, and absorption, that is what happens in the pathway. But also, we are going to be making reference to other functions like the endocrine, storage, defense function, and expulsion of the different undigested materials. Okay, and in this slide, you have a description of the different parts with the more specific functions that occur in each of them. Okay, we are going to be making reference also to the microbiota. Okay, these bacteria in the colon, for example, how they participate in the defense of the body, and also the importance that this microbiome has, for example, in preventing disease, in regulating appetite, satiety. Okay, something that is still under a lot of research, but is very, very interesting. Okay, there is nothing absolute about that, and it's very difficult to study, but we're gonna get some ideas about how this works. And today we are gonna be talking about, as I said before, a general overview of the GI tract, and you're gonna see the relationship between structure and function, and how there has to be a very well harmony between the different structures and between these structures and the nervous system for the body to be able to coordinate, integrate all of these digestive, secretory activity, the motility, and also all of the mechanisms of epithelial transport, okay, diffusion or active transport, primary or secondary, other types of transport that occur across the cells or in between the cells of the intestines, okay, how we manage to absorb what we need and not absorb what we don't need, and also how the body manages the absorption of water and electrolytes, okay, through the GI tract. So digestion is the first and most important step, okay, that will occur within the lumen of the GI tract. And this simply means the degradation, breakdown of different molecules of the food. And there are two types of digestion. Okay, we have one that is due to the physical action of the teeth, the muscles. Okay, we call that mechanical digestion. Okay, we break down the food, mix the food with different types of digestive secretions, for example, saliva, then the gastric juices, then the pancreatic juices together with the biliary secretions. And then we continue, okay, propelling all of this mixture, okay, throughout the alimentary canal or small intestine, okay, so we can perform the chemical digestion that is done by the action of the digestive enzymes, okay, there are different enzymes. Each of these enzymes has like a specialty. They break down specific things. So we convert all of these proteins, fats, and sugars or polysaccharides into monomers, tiny, tiny molecules that are the ones that are going to be absorbed. For example, amino acids from the proteins, fatty acids and glycerol from the fat, and monosaccharides from the sugars, from the carbohydrates or polysaccharides. And the secretions are extremely important. Without secretions, none of these would happen. Okay, we need, for example, the saliva to start breaking down the food in the mouth, and also for taste. Then we have the pancreatic secretions, the acid secretions in the stomach, okay, the gallbladder that will secrete. It's not secretion from the gallbladder, simply is the liver. We're going to talk about this at the end in the third encounter. Okay, bile is a secretion of the liver that is stored in the gallbladder. Okay, and then we have also the mucus that is produced by different mucosal glands at different areas of the GI tract. And this mucus serves to protect the mucosa, to prevent infection, to allow the lubrication of the GI tract, and to permit a better mixing of these food with different other secretions. And in this slide, you have the important fact about the GI tract, something that is very similar to what happens in the kidney, for example. We have a great intake of fluids during the day. Okay, we have the water that we intake, we have the saliva, gastric secretions, the biliary the bile that is produced by the liver, the pancreatic juices, intestinal secretions. So if you add all of these secretions plus the water intake, we have around 9 liters of fluids circulating in the GI tract daily. Okay, and we manage to absorb most of the water that is present there. So we normally eliminate only 100 milliliters of water with the feces, maybe between 100 and 200, but this is to represent the high efficiency of the fluid balance that occurs in the GI tract. Something very similar to what happens in the kidneys, okay, that we have lots of blood circulating through the kidneys every day. There is a lot of filtrate in the kidneys. Let's say an average in the healthy person of 100 milliliters of plasma per minute enter in the kidneys. And from that amount, only 1% ends up in the urine. So these are highly efficient absorbing or reabsorbing structures, okay, that if they fail, of course, we are going to lose lots of water. For example, when people have diarrhea or when people have, in the case of the kidneys, um, any condition like diabetes insipidus or something that is characterized by excessive water elimination. Besides digestion, we have the motility. Okay, motility is an extremely important property of the GI tract. That is going to be what we are going to discuss in the second encounter. And different areas have different types of motilities. Okay, and we are going to be studying some types of waves that occur in different areas. Okay, there are waves, for example, that maintain a very slow and very weak contractions permanently that will like clean the small intestine to prevent the bacterial overgrowth and clean all these to make it ready to receive more food for digestion. Then we have what happens at the sphincters. Okay, there are tonic contractions. Sphincters simply separate different areas, the mouth from the esophagus from the stomach from the small intestine, and the small intestine from the large intestine. And also we have some depolarizations that will produce other types of motilities. Okay, for example, these peristaltic movements and also the segmentation movements that occur in the GI tract. And absorption may happen through the cells, that is called a transcellular transport, or between the cells, and that is called a paracellular transport. Okay, we are going to be seeing what types of different things enter through the cells, transcellular or in between the cells in this paracellular route. And not less important, the defense function. I was saying before, our digestive system is one of the most important portals of entry, okay, together with the lungs, the skin. Okay, so the integrity of this mucosa and the proper functioning is essential as a defense function. Okay, we have epithelial cells that have very that have tight junctions, okay, making like a seal, creating a very good barrier. And also the secretions, for example, acid. Okay, it's very difficult for a virus or a bacteria to survive in that acid. Okay, there are some viruses that are classified as naked viruses because they don't have this lipidic envelope. These are more likely to survive, and these viruses are the ones that normally produce GI viral infections. But if the viruses have an envelope that is made of lipids, okay, they are not going to survive in the GI tract. Also, the enzymes have some antibacterial properties. The mucus contains a protein that is called mucin, and this mucin has antibacterial and antifungal properties. And the motility, of course, is very important. Okay, I was mentioning before, small intestine has this, even when it's empty, has this movement, okay, that is called migratory myoelectrical uh, currents, that this keeps the GI clean, preventing the bacterial overgrowth, guaranteeing that there is no leftovers in the GI tract. So all of these defenses belong to what we call the innate immunity, but we have also specific defenses, the T cells, the B cells, antibodies, this adaptive immunity. And we are going to be mentioning at the end the important role of the microbiome. But let's go to some more specific information. Okay, we have the function of the saliva. And sometimes we don't even think about it because we normally take these things for granted. But there are conditions, for example, some autoimmune conditions in which there is an attack against the salivary glands and some other exocrine glands. So these people don't produce saliva and don't produce tears. So imagine how complicated life is without saliva, how difficult it is to speak, how difficult it is to taste the food. And just imagine, maybe some of you have taken maybe Benadryl. Okay, Benadryl is a medication that will produce dryness of the mucous membranes. Sometimes Benadryl may appear in some over-the-counter cough medications or medications for the flu, etc. And you have experienced this probably. Or sometimes when you are talking for a very long time and you notice this dry mouth, how difficult it is to continue talking, because the moistening, the lubricating function of the saliva is essential for swallowing speech. Saliva is important as a defense mechanism. These people that I tell you with this autoimmune condition, in which they don't produce saliva, they have a lot of dental issues and infections, because the saliva is very important for preventing the bacterial growth. Helps in the formation of the bolus, is necessary for tasting, and also has a digestive action. There is an enzyme, the salivary amylase, that is the one that starts breaking down starch in the mouth. And here we have the regulation of the salivary flow, okay? And we are gonna be talking more about this when we talk about, well, we already mentioned the sympathetic, parasympathetic in nervous system, but when we talk about regulation of the GI tract, we are gonna go over these things. Notice that parasympathetic stimulation increases the salivary flow, but saliva production may also be increased by nausea, distension of the stomach, different foods, okay? And what reduces the salivary flow? Sympathetic nervous system stimulation, okay? Different hormones that normally conserve water, like antidiuretic. Sleep reduces the salivary flow. That's something that we have to be thankful for. And dehydration, of course, some drugs, like for example, Benadryl that I mentioned before, different sympathetic stimulating drugs, and also drugs that inhibit the parasympathetic nervous system. And aging. Aging is a factor that decreases almost everything that we need. So let's go to the secretions of the stomach, okay, gastric secretions. These are extremely important to understand, and it's a very good idea to try to create, okay, when you study, okay, try to read the chapter, read the topic, and then with everything closed, try to create a diagram, okay? Try to do this retrieval practice, okay? If you don't get it right, it's okay. The retrieval practice is what actually works on our brain, so we are able to retrieve the information when we need it, okay? The memory, some people say, the people who study these, the functions of the brain, okay, they say that memory is infinite. But the problem we have is trying to retrieve the information from memory. And there is something that works to improve that, and that is retrieval practice. Okay, simply doing, for example, the multiple choice questions that appear at the end, that appear in the book that you have, and just doing the effort. Okay, let me try to get this. Okay, looking at the question and not immediately looking at the answer, simply trying to do this effort to remember is what actually, okay, like opens pathways in our brain, so we are more likely to retrieve the information when we need it. So here we have the different cells, okay, that are present in the gastric glands. Okay, these are the glands that produce the gastric secretions that normally help in the digestion and also help lubricating, protecting the mucosa of the stomach. Okay, in the formation of the chyme, which is the product of the mixture of the bolus of food that we swallow with the gastric secretions. Okay, the cells in the gastric glands, there are stem cells that normally replace others that are damaged. Okay, there are cells that produce mucin, that are called mucus neck cells. Then we have the very important parietal cells. We are going to be focusing on these cells, are the ones that produce hydrochloric acid and also intrinsic factor. Then we have the chief cells that produce digestive enzymes, pepsinogen, and the gastric lipase. And we have endocrine cells that produce somatostatin and the mast cells that secrete histamine. We are going to see them in context when we see the regulation of the gastric acid secretion. The secretions are the acid, hydrochloric acid, that produces a very, very low pH in the stomach. Okay, when the stomach is empty, the pH there could be between 1 and 2. Okay, and it may go up to 4 or 5 when there is food present there. Okay, hydrochloric acid starts the digestion of the food there, also has antibacterial properties, and very important, activates pepsinogen to pepsin. Okay, every time you see this suffix ogen, that means inactive. Okay, this is the way the enzymes are made, and then the acid is necessary to break down pepsinogen and convert it to pepsin. That is the enzyme that starts breaking down peptides, okay, in the proteins, start the digestion of the proteins. Another important secretion of the parietal cells is intrinsic factor, okay, which is necessary to grab, okay, the vitamin B12 from the food, and then this intrinsic factor travels as a complex with vitamin B12, okay, to the ileum, the distal part of the small intestine, for this complex, okay, when it reaches there, allow the absorption of vitamin B12. We have then the gastrin, which is an enzyme, okay, that is produced by the G cells, G-gastrin. Okay, this is an enzyme, no, this is a hormone, okay, that stimulates the production of acid. Okay, so gastrin will stimulate acid secretion. Okay, we are gonna see a diagram where we are gonna see all of this integrated. But gastrin, besides stimulating acid, is also a growth factor. Stimulates the growth of the stem cells, stimulate the growth of the gastric mucosa cells, and also intestinal motility. Okay, has all of these functions. Okay, allows increasing acid and stimulates these mixing movements in the stomach, and also stimulates mass movements. Okay, there is a reflex that we are going to be studying that is called the gastrocolic reflex. And that means that when the stomach is full, gastrin will stimulate the colon, okay, for performing these mass movements and stimulating the defecation reflex. Mucus is very important as a protector of the mucosa. Okay, acid should never be in contact with the mucosa. There should be always a mucus layer in between. Okay, so mucus contains bicarbonate that will buffer the acid, protecting the mucosa, and also contains this important protein that is mucin. Okay, that is what makes the mucus thick, viscous, and has antibacterial and antifungal properties. And we also have to mention somatostatin, which is a hormone that inhibits many other hormones, and in this case will inhibit the function of the parietal cell, okay, by inhibiting, for example, gastrin. And then we have histamine, okay, that stimulates acid secretion, okay. And this is the diagram that I mentioned before. So we can see the regulation of the parietal cell, okay, that is producing the hydrochloric acid. Okay, this is a very simplified representation, but it's a very good one to understand how it works. Okay, normally these parietal cells, okay, are under the stimulation of the parasympathetic nervous system through the vagus nerve. Just thinking about food or the smell of the food, okay, will send signals to the stomach to stimulate these parietal cells so they start producing acid before the actual food enters. Okay, once the food is present and there is a distension of the stomach, then we have some local reflexes that will increase this production of acid even more. Okay, we have the parietal cell that normally secretes protons against a very, very, very high concentration. So we have to use an active transport, remember, against concentration. So the parietal cell is going to produce, and let me add some detail here, will produce protons, and also the parietal cell is going to produce or secrete chloride through another channel. Okay, and this chloride is going to go to the lumen, the hydrogen is also going to go to the lumen, and they are going to meet above the mucus layer. Remember, the mucus layer is protecting the mucosa, and there is a lot of bicarbonate. Okay, so lots of this hydrogen is going to be trapped by the bicarbonate, and just the hydrogen that goes out is the one that is going to react or bind to this chloride, forming hydrochloric acid. But this has to be away of the mucosa, because we don't want the acid touching the mucosa, because that will produce a lot of damage. And that's what happens in people who have acid peptic disease. Okay, that they have a lot less mucus, maybe because H. pylori infection, maybe because of them taking alcohol or taking ibuprofen or any other medication. But okay, this chronic exposure of acid to the mucosa will create ulcers, will create gastritis, and may even predispose to cancer. Now, how do we stimulate this structure here? That is the one that actually secretes or pumps the hydrogen. That's why we call it the proton pump. Okay, this can be stimulated by different factors. Acetylcholine from the vagus, or histamine that is produced by cells that are called enterochromaffin-like cells, or by gastrin that is produced by the G cells. Okay, notice that acetylcholine, the vagus nerve, may stimulate directly the parietal cells and also may stimulate the enterochromaffin-like cells to produce histamine. So we have 1 and 2 stimuli resulting from the vagus nerve during the cephalic phase, for example. And then we have gastrin. Okay, gastrin normally is produced when there is food in the stomach. Okay, the G cells are going to send this hormone through the blood. That's why we call it a hormone. And gastrin will directly stimulate the parietal cells, but also will stimulate these enterochromaffin-like cells. Okay, so when the food, just by thinking about the food, we are going to start producing acid from acetylcholine and histamine. And once the food enters, we are going to continue producing even more acid because of the action of gastrin and the added stimulation of these cells that will lead to production of more histamine. Okay, there you have the receptor for histamine. Okay, histamine receptors can be H1, that is normally present in the lungs, and H2 in the lungs and in the skin, for example, and in the mucous membranes of the nasal cavity. And the H2 receptors are the ones that are present here in the parietal cells. That's why we use medications that are called H2 blockers to inhibit these cells or this action of histamine. We also may use, for example, anticholinergic drugs like atropine to inhibit the acetylcholine action. Okay, or we simply can use proton pump inhibitors or PPIs. Okay, notice how we can create medications to act at different levels. Okay, the proton pump inhibitors, the H2 blockers, anticholinergics, all of them are going to work in reducing the acid production. And here we have a video, okay, but I'm gonna let you watch this video later. Okay, you have time for watching this video. We don't want to use time of the lecture for this. So we can advance. Okay, it's a very good explanation because reading sometimes doesn't give us an idea. Listening to someone sometimes give us a little bit, but when we see these animations and we see all this in motion, we get a better idea of these, and we can retain these for a longer time. Now, what are the phases of this gastric acid secretion? We already mentioned something about this. Okay, we have a cephalic phase, then a gastric phase, and then the intestinal phase. The phases 1 and 2, okay, will increase acid production, while the intestinal phase, the phase 3, will decrease acid production. Okay, the cephalic phase induces the release of a little bit of acid, let's say 10%, a very, very small amount. Okay, when we have the food in the mouth or thinking about food, someone talking about food or smelling food, okay, we produce saliva, but we also produce, okay, different, started increasing the production of mucus acid in the stomach. Okay, there are some mediators that are released, histamine, for example, okay, that will stimulate gastric acid secretion, but very little. Okay, once food is there, there is a distension of the stomach wall that induces a higher amount of acid production, let's say 90%. And what happens during the intestinal phase? Well, now we have the food or the chyme in the duodenum and jejunum. Okay, the presence of acid in the duodenum will inhibit the secretion of acid. Okay, acid reaches the duodenum. The duodenum produces a hormone called secretin, and secretin will stimulate pancreatic secretion of bicarbonate to neutralize the acid, and also will inhibit, okay, the secretion of acid because it will create a negative feedback on gastrin. Okay, there is another hormone that is called cholecystokinin movement, okay, that will promote biliary secretion and also pancreatic enzyme secretion. It's cholecystokinin promotes biliary contraction and the secretions of enzymes from the pancreas. Okay, different important hormones that participate in this phase. And this is like a summary, so you have it organized. Okay, these are different factors that stimulate acid. Okay, vagal activity, histamine, gastrin, also insulin. That means we take sugar, sugary drink, insulin increases, and insulin increases acid. Okay, that way we understand why when we take something that is very sugary, we may have symptoms of GERD, okay, or some discomfort in the stomach. Caffeine as well, stress, too much coffee, too much stress, too much sugar, not good for the stomach. And this acid can be inhibited by somatostatin. Somatostatin will inhibit gastrin. Okay, then we have some other peptides, for example, GIP. Okay, GIP has different names. Glucose-dependent insulinotropic peptide. Okay, this occurs when food is in the duodenum or jejunum. Secretin from the duodenum that also inhibits gastrin, and peptide YY from the ileum and the colon. So even the presence of food in the distal small intestine and in the colon may inhibit the production of acid. And here you have a chart. Okay, and I recommend you, every time you see this chart, try not to simply say, oh, this is great, and I'm going to use this for studying. I recommend you to, of course, study, but try to make it yourselves. Try to create it. Okay, I'm going to focus first on gastrin. Let me try to reproduce it without looking at it. Okay, the same thing that I was telling you before, retrieval practice. Or you can make, for example, flashcards and practice this retrieval. Okay, that trains the memory, trains the brain. There are different things that are in bold letters. These are the most important ones. Okay, gastrin, G cells. Okay, the stimulus here you have different stimuli, stretch of the stomach, vagus nerve, increases the gastric hydrogen production. Secretin increases bicarbonate secretion. Okay, increases biliary and small intestine buffer secretion. Okay, the biliary juices, the bile has a very alkaline pH too. Okay, so will neutralize the acid. Cholecystokinin, produced by the duodenum jejunum, okay, will be released under the stimulus of different peptides, amino acid, fats present in the duodenum, increases the pancreatic enzymatic secretion, enzymes from the pancreas. And also cholecystokinin, kinin means movement of the bladder, gallbladder, contracts the gallbladder and relaxes the sphincter to allow the emptying of the gallbladder. Also produces satiety. Okay, when we eat, when the food is present there, okay, also will increase the GI tract motility, so will stimulate, for example, defecation, will reduce the gastric emptying, because cholecystokinin will guarantee that we mix very well all the chyme with the bile and the pancreatic secretions and send it, okay, to the rest of the small intestine. And when the duodenum is empty, then another portion of the contents of the stomach enters into the duodenum. Okay, it's like regulating that the emptying of the stomach occurs slowly, sequentially, and organized, and is organized. What other things? Well, we have this GIP here, okay, that decreases the gastric acid production, increases insulin. That's why this GIP is called an incretin. Incretin. Okay, this stimulates insulin secretion. before the sugar enters into the blood. Okay, it's like preparing the pancreas. Oh, there is sugar in the food. Let me tell the pancreas that the sugar is coming. Pancreas produces some insulin before the actual insulin sugar enters in the blood. For example, if you put IV glucose, okay, the glucose levels don't start going down until some minutes, until after some minutes, because of the absence of this incretin effect that only occurs when sugar is in the duodenum and jejunum before going to the blood. Other hormones are motilin. The name is self-explanatory. Motility increases the motility. Okay, and then glucagon lipeptide 1, GLP, GLP-1, that is another incretin together with GIP and produces satiety. It's a very good chart. Try to please study this very well. Well, I think here we already mentioned the function of secretin and CCK. Okay, so there is nothing new here. What we wanna talk about is about the enzymes of the pancreas. Okay, and some important information that you need to understand this and what happens later when there is a pancreatic insufficiency. Okay, the main function of the pancreas is to buffer the acidic chyme. Okay, the pancreas produces lots of bicarbonate. Okay, once the chyme enters into the duodenum, has a pH, let's say, of 4, more or less, 4 or 5, depending on the composition, depending on the amount of gastric juice. Okay, we need to add bicarbonate. Bicarbonate will inactivate the pepsin that is there. Okay, will change the pH, will neutralize this pH, making it ready for the movement, okay, along the GI tract, small intestine, large intestine. And this neutral pH is what will allow the activation of the pancreatic enzymes. Pancreatic enzymes don't work if the chyme is acidic. They are not going to be activated. Okay, so this bicarbonate is essential for digestion. And if people don't produce enough bicarbonate, or if they produce too much acid, so we don't achieve the neutral pH, the digestion is not going to occur, and the food is going to continue, it's not going to be absorbed, and it's going to be eliminated, okay? And people are going to have diarrheas that we call malabsorptive diarrheas that contain lots of fat, for example, and people develop nutritional problems as a result of this. Which are the enzymes? There are some proteases to break down proteins. We have amylase, that is very similar to the amylase from the saliva. And we have lipase to break down lipids. Okay, the proteases are trypsin, chymotrypsin, and elastase. Okay, they are made as zymogens. Notice the name, trypsinogen, that has to be converted to trypsin. Chymotrypsinogen, inactive, that is going to be converted to chymotrypsin, and proelastase, which is the one that later is activated to elastase. Okay, in these zymogens, always try to apply the same concept. Okay, remember this system, that renin-angiotensin-aldosterone system, that renin breaks down or activates angiotensinogen from the liver and converts it to angiotensin 1. It's the same principle. Okay, trypsinogen to trypsin, angiotensinogen to angiotensin. Okay, they are activated, these zymogens are going to be activated by the brush border enzymes. Okay, when you enter into the duodenum and rest of the small intestine, you know that we have this villi, and the villi have this microvilli or brush border. Okay, in between these villi, microvilli, there are enzymes that are made by the cells of the small intestine. For example, enterokinase. This is the one that breaks down trypsinogen to convert it to trypsin. Okay, so if there is any damage to the mucosa, for example, people with celiac disease, they are not going to have the enterokinase working. So these zymogens are not going to be activated. That's why they also have malabsorption, okay, and as a manifestation. Diarrhea, fat. Lipase is the one that breaks down lipids, amylase, digestion of carbohydrates. For example, starch is converted to maltose. Maltose is a molecule that contains two molecules of glucose, glucose plus glucose. So it's a disaccharide that has to be converted to a monosaccharides. And triglycerides are going to be broken down, okay, by lipase, okay, into free fatty acids and monoglycerides. A triglyceride is a molecule of glycerol with three fatty acids. So what this lipase does is separates one fatty acid, another fatty acid, and the glycerol with one of the fatty acids, and that is called a monoglyceride. This digestion requires that the lipids are coated with bile. This process is called emulsification. Okay, and there is another factor that is necessary, and that is colipase. Colipase is a cofactor for the lipase, and that will displace the bile, allowing lipase to enter to do the digestion. So they have to work together. These are the fat molecules surrounded by bile. Lipase is waiting. Lipase can't enter through the bile, so we need the action of colipase that opens the door, separates the bile so the lipase can enter and have access to the fat. It's very interesting, this process. And then we have the action of the hormones. Okay, I think all of this is in the table. I think all of them are there, so we don't need to go over them. Yeah, all of this is in the table, so I don't think we necessarily have to enter there. Please, any questions before going to the break, okay, about these things? It's a lot. I know it's a lot. So if you have any questions, let me know now. If not, you think about questions and we can discuss them during or after the break. Okay, perfect. Thank you so much. Okay, let's see each other in 10 minutes. Okay, it's 10:52 at 11:02. Okay, thank you so much. See you soon. Okay, we are ready to move to the second part. And well, we have to talk about this part of the autonomic nervous system. We already mentioned this when we were covering the nervous system, and here we are going to see some connections, okay, between what we studied, okay, and how this part of the nervous system works. As I told you, there are a lot of uh, debates about if the enteric nervous system should be considered a part of the autonomic nervous system or should be considered a part. Okay, this is a local network of neurons that contains as many neurons as we have in the spinal cord that normally regulates the function, motility, and secretion of the GI tract, okay, by means of reflexes, okay, without any input from the central nervous system. Now, it is under the control of the sympathetic and parasympathetic, and here is when, or the point where many people don't agree. It is part or it is not part of the autonomic nervous system. So we're not going to enter into that debate or try to solve that. We are simply going to understand how it works. Okay, there are reflexes as the reflexes that we have in the spinal cord. There are afferent neurons that normally are monitoring the different conditions, stretch, the contents, the pH, for example, and some interneurons that will integrate this information and then will send information through efferent neurons that will either stimulate or inhibit the motility, the secretion, okay, increase the absorption, or participate in immune functions. It's important to know that these afferent neurons, these sensors, are not only sensing the content of the food, also the type of bacteria, for example, that is present there. Bacteria release different chemicals as a result of their metabolism, and this uh, nervous system, the enteric nervous system, reacts to these different bacteria or the products they produce. That's why the balance between different types of bacteria is so important in our gut. Okay, there are also some, not only neurons, there are neurotransmitters, okay, the ones that we mentioned before, that also participate in this process. Okay, this enteric nervous system is connected to the central nervous system, okay, in what we call the brain-gut axis. Okay, very important connections that are sometimes complicated to study. And then in the case of proximal structures, okay, for example, the esophagus, stomach, it's a good idea to understand that they are under the central nervous system control more than distal structures. And that's not difficult to understand. For example, the area or the function of swallowing, okay, the first part of taking the food into the GI tract is something that is more controlled by skeletal muscle, even voluntary actions, the swallowing, for example. Okay, the first third of the esophagus contains skeletal muscle, so it's under the control of the somatic nervous system, and then the number of smooth muscle fibers increase little by little until at the end of the esophagus we have only smooth muscle. So it's like the central nervous system, the somatic part, is giving away the control of the digestive function to the autonomic nervous system little by little. It's something interesting how this is designed. This diagram shows, okay, the different components of the enteric nervous system. Okay, it is basically composed of two plexuses or plexa. Okay, one is called the myenteric. Okay, that is located between the longitudinal and the circular layers of the GI tract. Okay, remember there is an outer longitudinal and inner circular. So in between these two, okay, we have the myenteric plexus. And then we have the submucosal, okay, that is under the mucosa. It's between the mucosa and the circular muscle layer. So it's closer to the lumen of the GI tract. Okay, you have some sections here. So you see how it looks like, okay, when we look at it under the microscope. Okay, they function, as I said, without the intervention of this sympathetic parasympathetic nervous system, but in some occasions it is necessary the intervention, okay, of these two systems. Okay, notice here the connections between the parasympathetic, okay, from the vagus nerve and from the pelvic nerves, okay, and the sympathetic division, thoracolumbar origin, okay, that also participates in the regulation when it is necessary. Okay, here you have another diagram, but that more or less it shows the same. I wanted to include this here, okay, so it serves as a review of what we covered last week. And we were talking before about retrieval practice, okay, and one important thing for studying and guarantee long-term retention of the material is that this retrieval practice is done spaced in time. Okay, I know many students, and that is totally normal, they use a technique for studying that is called mass uh, review of the material. Okay, for example, you have a test, pharmacology of the antiarrhythmic drugs. And maybe you spend 4 days studying this, but that's great for the short-term retention, but it's not good for the long-term retention. Ideally, we should study, okay, different things, or for example, study the autonomic nervous system every, let's say, 8 days or 10 days. Spacing, spaced repetition, and retrieval practice of this material, the important concepts, not everything, is what guarantees the long-term retention of these things. So you can review here the different things, okay, about the sympathetic, parasympathetic nervous system. And here we have the effect of the major gastrointestinal hormones on the GI motility. Okay, so this is something that we already saw in the chart, okay, before. So you have here the diagram, okay, that shows, instead of in a chart, okay, it's a graphical representation in which you can see the connections, okay, between all of these structures and the action, or trying to give you the big picture of this control by hormones of the GI motility. So let's go to the immune functions, defense functions. Okay, that is a very important structure that is called the firewall, mucosal barrier firewall, that interacts with the microbiome, okay, the normal flora. Okay, there are different components that are described here. First of all, the mucus layer. Okay, this thick mucus will trap any bacteria. Okay, if any bacteria enters there, we have the mucin that has antibacterial properties. Okay, this prevents what we call microbial translocation, or movement of microbes down the barrier into the submucosa. We should avoid that. Okay, the epithelial cells produce different peptides, defensins, lysozyme, mucins. Antibodies are also released. Okay, and these are very good antimicrobials. Okay, then we have different processes that occur if a bacteria enters into the submucosa. If there is any bacteria that translocates, okay, we have under the mucosa the macrophages, phagocytic cells. They can be also captured by lymphocytes, and we have the actions of plasma cells that produce antibodies. SIgA stands for secretory IgA. immunoglobulin A that will go and will trap, neutralize any bacteria, viruses that might be present there. Okay, there we have also a representation of the different cells that may be found, okay, below the mucosa. Okay, in the small intestine, we have very important components of the immune system. We have the Peyer's patches. We have lots of lymphoid tissue below the mucosa. Notice how many components are there: lymphocytes, B and T, plasma cells making antibodies, phagocytic cells, mast cells that will release histamine and promote vasodilation and inflammation, and also eosinophils that are important to fight parasites. Okay, there you have the integration between the function of the intestine and the lymphatic system. Okay, besides the Peyer's patches, we have also lymph nodes located in the mesenteries. Okay, if any bacteria, virus enters in the lymphatics, they are going to be trapped there. Okay, they are going to be processed there, and we expect that they don't enter into the circulation. Okay, because if they do, we are going to have septicemia, we are going to have a more generalized infection. It's important to understand how all of this is made, how it works, what is the connection, what is the interaction between different facts. Sometimes when we study, we obtain like isolated amounts of knowledge. Okay, we study, for example, lymphatic system, we learn this. Digestive system, we learn that. Circulatory, we learn, but sometimes it's difficult to make the connection between the different things. Okay, and that's why it's important, this integration of the different things that you learn in different systems. Well, we have to make reference also to something that is called the splanchnic circulation. Okay, notice splanchnic circulation refers to all the circulation, arterial and venous, okay, that provides blood to the different structures of the abdominal cavity. Okay, we have some arteries, veins are called mesenteric arteries, mesenteric veins. And notice how the branches from all of these veins, for example, superior, inferior mesenteric veins, together with branches from the stomach and spleen, and from the lower part of the esophagus, all of them are going to enter into the portal vein. Okay, so everything that is absorbed in the small intestine, large intestine, and the secretions or the different things, not the secretions, the circulation from the pancreas, spleen, stomach, etc. All of these things are going to travel directly to the portal vein. And for example, carbohydrates, lipids, proteins, etc. are going to enter the liver to be metabolized. And from there, it's going to continue traveling. We're going to talk more about this specifically in the third encounter. Okay. Not only food, also medications that are absorbed are going to enter and are going to be metabolized in the liver. So let's take a look at a brief overview of digestion. Okay, we are going to be talking about digestion of carbohydrates, lipids, proteins. Okay, carbohydrates, sugars. There are different types. Okay, one very important is glycogen. Okay, that is, starch that is present in many carbohydrates, potatoes, rice, etc., bread. And then we have others, for example, lactose from dairy products, sucrose or regular sugar, cane, etc., or beet sugar, carbohydrates, and mostly starch. Okay, the digestion of it is going to start in the mouth. Okay, the salivary amylase starts the digestion of starch, and you may make an experiment. For example, you can take a piece of bread and put it in your mouth and let it there for a while, and you're going to see that after some minutes, the bread starts being dissolved and starts tasting sweeter because this salivary amylase starts breaking down, okay, the polysaccharide into disaccharides that stimulate more with more intensity the taste receptors. Starch is going to be converted to a disaccharide that is called maltose. And we also take with the food other types of disaccharides, for example, sucrose and lactose. Okay, so these disaccharides, after ingestion, have to be broken down by enzymes called saccaridases that break down the disaccharide into two monosaccharides. For example, maltose is composed of two molecules of glucose. It's going to be broken down into glucose and glucose. Sucrose is composed by glucose and fructose. Okay, we have the enzyme sucrase that breaks it down into glucose and fructose that are going to be absorbed. Lactose is another disaccharide composed of glucose and galactose. And then we have the enzyme lactase that breaks it down. Okay, you have here like a summary of where the different enzymes are, for example, where they act, and their substrate, they act on polysaccharides like starch. And what is the end product? Okay, maltose for starch. And the saccaridases that are present, located in the brush border of the small intestine. Okay, their substrate, they act on disaccharides, and this is the end product. The monosaccharides that are the ones that are going to be absorbed. Okay, we don't absorb lactose, we don't absorb sucrose, we don't absorb maltose or glycogen. We don't have that capacity. And this is the same thing, okay, but this is represented in this diagram. You have the different sugars that we take, okay, and how different enzymes, salivary, pancreatic amylase, start breaking this down. And here we have the disaccharides. Okay, notice that the digestion of the disaccharides occurs in the small intestine brush border by the epithelial cells enzymes, these lactase, sucrase, maltase, that break down these. And we obtain the monosaccharides, glucose, fructose, and galactose that are going to enter, going to pass through the epithelial cells to the blood, and from there it's going to go to the portal vein, liver, and the liver is going to take care of processing or storing or using these monosaccharides. In the case of proteins, something similar happens. Okay, we start the digestion of the proteins in the stomach. Pepsinogen, okay, is converted to pepsin. Remember, acid is necessary for that. Pepsin starts the digestion of the proteins there, and then the proteins in the chyme enter into the duodenum, and they get mixed with the pancreatic enzymes. Okay, trypsinogen is going to be converted to trypsin, chymotrypsinogen to chymotrypsin, and proelastase to elastase. For this process, okay, we need the neutralization from the pancreatic secretions. Okay, the pancreas, that is a typo there, not pancreases. Pancreas, okay, secretes an enzyme that is called trypsin inhibitor, okay, because we want to prevent, okay, the premature activation of trypsinogen. Okay, let's say this is the pancreas, and that is important to understand for later pathophysiology. This is the pancreas, this is the pancreatic duct. Let's say trypsinogen is traveling here. Okay, the pancreas produces also this inhibitor, so trypsinogen doesn't get activated inside the pancreas, but only because, remember, this fluid here is alkaline. Okay, so it could allow the activation. So once this trypsinogen is released, then, okay, we have the activation. Who activates trypsinogen? Enterokinase. The first step in the activation of the pancreatic enzymes is activating trypsinogen to trypsin, and then trypsin is going to activate the other two. Okay, it's a very well designed process. Okay, we shouldn't let trypsinogen get activated here just when it reaches the duodenum by the action of this enterokinase. Then trypsin will activate this and this. And also there are some intracellular peptidases, but that is a topic of cellular biology. And here we have the representation of this. Let's say we are eating meat. Notice the presence of intrinsic factor B12 action here. Acid activates pepsin. Notice the complex between intrinsic factor and B12 traveling alone. Now we have some peptides after the action of pepsin traveling to the duodenum. We have the secretion of the pancreatic enzymes, the zymogens, and then we have the action of enterokinase activating trypsin first, and trypsin activating the other ones. Then we have the polypeptides that are going to be broken down by other enzymes that are in the microvilli, in this brush border, and the amino acids, single amino acids being absorbed. Some of them are going to enter the portal circulation. Some others may enter the thoracic duct, but most of them are going to enter the portal circulation and will continue to the liver. Now, lipids, this process is a bit more complex. Okay, we have lipases in the tongue and in the stomach, but these are not too important for lipid digestion. Okay, they may have a more important role, for example, in protecting our body against bacteria. Okay, bacteria have a lipidic component. Viruses may have a lipidic envelope. So these lipases may help eliminate excess bacteria, viruses, etc. The most important enzymes in the digestion of the lipids are the pancreatic lipase and the colipase. We already mentioned that, how colipase opens the door. So lipase can enter through the bile and digest the triglycerides. Now, notice the action of trypsin. Remember, trypsin is very important to activate the rest of the enzymes of the proteases. Trypsin is also important to activate procolipase. Okay, let's go back to our pancreas. The pancreas is producing the zymogens, trypsinogen, and also produces the procolipase that will help lipase in the digestion of the fat. We don't want the pancreas to be digested. We don't want anything that destroys the pancreas. So lipase could digest the pancreas if colipase was helping it. Okay, so we don't want these two to get activated inside the pancreas. That's why the pancreas produces the inhibitor for trypsinogen, and trypsinogen will be activated in the duodenum to trypsin, and trypsin will activate all the other proteases and will activate the procolipase. So notice that this chain is necessary for proper digestion, but more importantly, to prevent the damage to the pancreas by the digestive enzymes. Okay, now these are the enzymes. What happens with the fat, triglycerides, cholesterol, etc.? They must be incorporated into structures that are called micelles. Which are micelles? These are fat molecules surrounded, okay, by biliary salts, bile salts. okay, to make very tiny droplets of fat, okay? Then we have the action of these lipase and colipase breaking down the triglycerides into free fatty acids and a monoglyceride, FFA for free fatty acids and a monoglyceride. Okay, that's the way we absorb the fat. Once the fat is inside the cells, the two fatty acids and the monoglyceride are rearranged into a triglyceride. And the triglycerides are going to be packed into chylomicrons. Okay, chylomicrons are the ones that enter into lacteals that are inside the villi. Inside the villi, we have capillaries from the arterial circulation, venous capillaries, and also we have lymphatics. These are called lacteals. Okay, so the chylomicrons are going to enter the thoracic duct, lymphatic system. I'm sorry. Are going to enter the lymphatic system, okay, and from there are going to enter the subclavian vein. Notice that the fat, most of the fat doesn't enter the liver, goes directly to the systemic circulation. Okay, proteins, amino acids, and glucose and monosaccharides go through the liver, but fat bypasses the liver and goes to the systemic circulation. Some exception, okay? There are some medium chain fatty acids that may go to the liver, but most of it enters the systemic circulation. And here we have this, okay? There you have, notice that this is more complex. Okay, we have the food or the different lipids, triglycerides, diglycerides, monoglycerides, fatty acids, cholesterol, different types of fats entering the duodenum. There is a mixture with the pancreatic juice and the bile. Notice this structure here. Okay, these micelles, sorry, this is all the fat that is going to be surrounded by these biliary salts. There has to be emulsion first, so we divide the total amount of fat into tiny droplets surrounded by the biliary salts, and these are the micelles. Okay, we have the action of the lipase, colipase. Okay, here we have the breakdown of the triglycerides into monoglycerides and free fatty acids. Okay, and these tiny molecules are the ones that are going to be absorbed through the apical part of the enterocyte. Okay, and they are going to be packed into the chylomicrons. Okay, and most of these chylomicrons are going to enter the lymphatic circulation. Okay, some tiny molecules, the medium chain fatty acids, may enter without being packed into the portal circulation to the liver. Okay, but most will take this pathway. The same pathway is going to be taken by vitamins that are soluble in fat, A, D, E, and K. Okay, they are going to be absorbed together with these triglycerides, etc. So we have to talk about vitamins, okay? Vitamins, or every time I say the word vitamin, I remember my professor of biochemistry. He's a very good, amazing professor from Liverpool in England. And the first day of class, he said, I know that you say vitamin, but I am from Liverpool and the correct form is vitamin. So if you want to keep an A, if you want to keep a good grade in my course, you better say vitamin. Okay, professor, as you say. I was a very, very nice person. So Vitamins, this word comes from the union of two words, vital amines. Okay, this is the origin because they are essential for our life. Okay, these are organic compounds that are required for the metabolism of many different things. Okay, and we cannot synthesize vitamins, so we must ensure a constant, steady supply. The only exception, well, a few exceptions, but vitamin D is the most important. But in fact, vitamin D is every day more considered a hormone rather than a vitamin. Okay, it has a similar structure to some vitamins, but is more a hormone made from cholesterol, like many other hormones, for example, aldosterone, estrogens, testosterone, they are made from cholesterol. So exactly in the same way we make from cholesterol vitamin D. So it's kind of more hormone than a vitamin. What are the sources? Most of them are acquired from the diet. Okay, vitamin D is synthesized by the body, but of course we also can get it from the diet. And vitamin K, that is produced in some amounts by the intestinal bacteria. Okay, the vitamins are called micronutrients together with the minerals, important minerals of the body, simply because they don't provide energy. Okay, this name micronutrient is misleading. Some people think, oh, it's a micronutrient, we don't need that. No, we don't do anything with the macro if we don't have the micro. Because, for example, in order to make ATP from glucose, we need vitamin B. Okay, so we don't do anything with giving glucose to a person if the person doesn't have vitamin B in the body. It's not going to use the glucose. Okay, they have a very specific and critical biochemical roles. That's why deficiency or excess may produce some diseases. For example, some of the vitamins act as coenzymes, B vitamins, A, K, or antioxidant action, C and E, participate in cell signaling for vision, for example, vitamin A, help in gene transcription, vitamin A and E, and act as hormones, vitamin D. Okay, vitamins may be classified depending on the solubility into fat soluble that are absorbed with the fat, and the body stores them in the, for example, in the adipose tissue or in the liver. Okay, or water soluble. Okay, normally we don't store the water soluble vitamins except for two of them, folate and B12, cobalamin. Okay, there are different differences. I've read this topic in many different sources, and they all say something different. For example, uh, some books say that we have folate enough for 6 months, and B12 for 1 year. Okay, other books say we have folate for 3 months and cobalamin for 3 years. So there is a lot of disparities in this. But the important thing is when someone has nutritional problems, they are going to develop folate deficiency first and B12 deficiency later. Okay, let's say we can accept the 6 months and 1 year, but that doesn't have to be too absolute. I mean, maybe it depends on how much people have before they started having the issue. Okay, it's like if you retire and you have only $1,000 in your uh, bank account, that's going to be a very important difference with someone that has a 401k with some tens of thousands. Yes. There may be different deficiencies. For example, malnutrition, malabsorption disorders, restrictive diets. For example, a strict vegan diet in a person who doesn't take supplements may develop maybe 1-2 years after they start a B12 vitamin deficiency. Also, some of them may produce disorders because of overdose, excess. But this is very specific for the ADEC vitamins because are the ones that can be stored. If I decide today to take, for example, 10 grams of vitamin C, the only thing I will develop is gastritis because the excess vitamin C is going to be eliminated by the kidney. But if I decide to take an overdose of vitamin A, I may start having vision problems. Okay, these vitamins are going to be stored in the adipose tissue, so I'm likely to have issues. These are the different vitamins, the water-soluble ones, that you have the names, ascorbic acid, the C, and they have the different B vitamins, thiamine, riboflavin, niacin, pantothenic acid, pyridoxine, folate, biotin, cobalamin. Okay, there you have some, is exactly what we mentioned before. These are water soluble, so they are easily taken by the cells, except B12 that, as we already mentioned, intrinsic factor is required for the absorption. And then we have the... I found this on the web. No, I didn't ask you anything. And the same thing happens with the storage. They are not stored except B12 and folate. I forgot to add B9. They are stored in the liver. And the fat soluble, we have the vitamin A, also known as retinol, very important for vision, antioxidant, gene transcription, calciferol, which is vitamin D. Calciferol is made from cholesterol. Okay, very important for the calcium metabolism, phosphate homeostasis. Okay, tocopherol, which is vitamin E, an antioxidant. And then we have vitamin K. Has different names, but I chose to write the name of the active one, okay, which is hydroquinone. Important for the activation or for the production as a cofactor for clotting and anti-clotting proteins. And they are well digested, absorbed in the same manner as the lipids, and they are stored in liver and in the fat cells. And something that is important to know, for example, let's say someone has, takes lots of vitamin A for a lot of time, okay, and they start developing, let's say someone takes vitamin A in excess for 1 year, and they start developing vision problems, because the deficiency and the excess vitamin A will produce more or less the same. And this person seeks for medical attention, and the clinician tells them, you have to stop taking the vitamin A. Well, that doesn't mean that they are going to get better immediately, because our body will start releasing all the vitamin A that is stored in the fat cells, and it's going to be little by little, okay, releasing, and we have to wait a lot until these stores are depleted. The same happens, for example, with medications that are fat soluble. Someone takes an overdose of a medication that is fat soluble. They stop taking the medication, but the fat tissue, the adipose tissue, continues releasing the medication for a while. It's not the same with the water-soluble medications that are going to be eliminated immediately by the kidneys when they are in excess. But this is more or less the same thing that we mentioned before about the absorption, the processing and absorption of lipids. Okay, micelles first, then chylomicrons through the lymphatic vessels. But here we are adding the detail of the vitamins, okay, that are also absorbed and incorporated into the chylomicrons to be to enter the body through the lymphatic system. Okay, absorption, incorporation, and circulation. Well, I'm going to make a little reference to the metabolism of vitamin D. Okay, remember this is made from cholesterol. Okay, we also obtain some from the diet. Okay, the first step is going to be the formation of what we call vitamin D. Okay, then we also have vitamin D3 from the food. Okay, this vitamin D or cholecalciferol, okay, is gonna be processed in the liver. There is an enzyme that is called 25-hydroxylase that converts vitamin D3 to calcidiol. Okay, and then this calcidiol or 25 vitamin D, the easy name is 25 hydroxy vitamin D. Okay, this one is going to be activated at the level of the kidney by another enzyme, 1-alpha hydroxylase, stimulated by parathyroid hormone, okay, into calcitriol or 125 dihydroxy vitamin D. Okay, the way that I have to remember this, because we have to use mnemonics, and we want to remember lots of information. Mnemonic is a help for the memory. Okay, the way I remember this is, okay, this is the first step, this is the second step, and this is the third. Notice that this is calcidiol dual di 2, second step. And this is calcitriol, like 3, third step. Okay, calcidiol, and then calcitriol is the active form, the one that is activated in the kidney. And well, here we have the absorption of the vitamins. Okay, these are the water-soluble ones. Notice that there are some different transporters for different enzymes. They are mostly absorbed at the level of the jejunum. Okay, and then, for example, the B1, B2, B6, they are transported. They have a specific transporter. Folate should be absorbed together with protons, with hydrogen, and the other ones, niacin, etc., vitamin C, they are going to be absorbed together with sodium in this type of transport that is called a secondary active transport. Okay, this sodium enters, is going to be taken out of the cells by the sodium potassium pump. When the concentration of sodium here is very low, it will drive more sodium and vitamin C, for example. This is the same mechanism, for example, that our intestine uses to absorb glucose and to absorb amino acids. Glucose and amino acids should be absorbed together with sodium using this secondary active transport mechanism. So we are not going to dedicate much time to talk about all the minerals. That would be impossible. But it's important to talk about iron, which is extremely necessary for hemoglobin and oxygen transport. Okay, iron, okay, is absorbed, well, from the diet, of course. Okay, it is necessary the acidity of the intestinal lumen, okay, for absorption in the duodenum. So that's why, for example, when people take iron supplements, they should take them with something more acidic, like orange juice, and not with milk or anything that creates an alkaline pH. For example, people taking PPIs or anything that, or antacids, will have a lower iron absorption. Okay, the absorption of iron is regulated at the level of the duodenal mucosal cells by an enzyme that is called hepcidin, that is produced by the liver. For example, when there is inflammation, the liver makes hepcidin that will inhibit the iron absorption. Okay, iron enters the duodenal cell mucosa cells. It's going to be bound to a protein that is called ferritin that allows the storage, can stay there. And if necessary, iron is going to be absorbed. Okay, there is a transporter that is called ferroportin, port, entry. Okay, iron will enter if hepcidin is not there, okay, and will be bound to transferrin, that is the one that transports the iron in the blood, okay, may be bound or and will this iron will travel bound to transferrin, and it's going to enter the cells, for example, in the liver or in the bone marrow, in whatever organ, and it's going to be used, okay, for making hemoglobin or simply to be stored, okay, in the ferritin that is present also in other cells. Okay, but the regulation by hepcidin is very important. Inflammation produces hepcidin, the liver produces hepcidin that inhibits the iron absorption. That's why people, when they have chronic inflammation, they develop anemia, specific anemia that is called anemia of chronic inflammation. And well, this is representing more or less the same, okay, the transport of iron using transferrin. Okay, you don't need, of course, to memorize any numbers here. Okay, simply understand iron is transported using transferrin to the liver back and forth. Okay, part of it is going to enter the bone marrow to make red blood cells. Okay, there is nothing new here. And we're going to be talking about calcium and magnesium. Okay, calcium is a mineral, extremely important, the most abundant mineral in the body. Okay, that is going to be absorbed in the duodenum mainly. And for the metabolism of calcium, we need the action of vitamin D, the active form, the calcitriol, 125 dihydroxyvitamin D, okay, that will regulate the channels, okay, for the absorption of calcium, and also intracellular proteins that will bind to calcium and allow the export of calcium. Okay, calcium is going to enter through a channel, it's going to bind to a protein, and it's going to be exported out by calcium ATPase. Okay, all of this is stimulated by vitamin D. So the action of vitamin D is to stimulate calcium absorption. And there are other mechanisms that are independent of vitamin D that simply depend on the concentration gradient. Okay, but this calcium ATPase is the one that pumps calcium against the concentration gradient. So when necessary, we absorb more calcium, doesn't matter how much we have. And magnesium is a mineral that every day we understand better the importance. And magnesium is one of the most important intracellular minerals. Okay, we are going to be seeing in the future the importance of magnesium for the thyroid function, for example. Is absorbed throughout the GI tract entirely and depends on dietary intake. Okay, there is a channel that normally is upregulated when there is low magnesium. Okay, so this is the GI tract. Let's say someone has normal magnesium level and they have 100 channels, just to say a number. When people have low magnesium, our intestine cells are going to upregulate the number of channels. Let's say instead of 100, now we are going to have 1 million. So we absorb a lot. If we have too much, then we are going to down regulate the channels, so we are not going to absorb too much. That's the way magnesium absorption is regulated. Well, I think we have some slides that we better continue on the next encounter. Okay, we are almost at the end, and I really don't want to overwhelm anyone. And I think that it's very important that you have a fresh brain before going to the part of regulation of the food intake and microbiome, etc. Okay, if we simply try to rush, okay, through these slides now at the end, it's not going to be beneficial. So I wanted to ask if there is any questions before saying goodbye for the day.
Dr. Deano, will the remainder of these slides then be included with the recording for Wednesday?
If you are aware, we are going to have a long session. We're going to have lecture from 3 to 5 and then the TBL from 5 to 7. That's the last information I got. That means that I know you're going to be studying for the exam on Wednesday, but remember there is going to be also a TBL at 5. Okay, so review the different things that we saw today, okay, in preparation for that TBL. I don't know, maybe you have more information than, no, not for the quiz on the TBL. I think you have more information than I have.